Hello everyone, welcome to Sudha's classroom. Today's class we are going to discuss about the proteins and its qualitative analysis. What are proteins? Proteins are biomolecules formed by the repeated linkage of amino acids. So this contains peptide bonds. Proteins are very essential for our body because it maintains the structure, regulation and functions of every cellular process in our body. So when you see about the proteins, it is found in plenty of foods like meat, egg, pulses, nuts, etc. In today's session, we are going to see about the qualitative test of proteins. There are a lot of qualitative analysis for proteins. So here, we are going to discuss about the main test, that is common test like Biuret test and Ninhydrin test. So first, let's use the example. Actually, it is an unknown sample, but we are using as the protein sample from the egg. So we know that proteins, first we have to test the solubility. So to the small quantity of the protein sample, we are adding the cold water. The protein sample is insoluble in the cold water. Now, we are testing the solubility using the hot water. To about 1 ml of proteins, we are adding boiled water. It is partially soluble in the hot water. The next test is about the Biuret test. So here, to the small quantity of protein sample, like we can take around 1 ml of protein sample, we are adding the Biuret reagent. The Biuret reagent is a mixture of copper sulfate and sodium hydroxide. We can see the violet color formation, which denotes the presence of peptide bonds. So proteins are made up of peptide bonds, and this Biuret test confirms the presence of peptide bonds. Copper sulfate present in the Biuret will react with the peptide bonds present in the protein sample, and this will form the color violet color. So this denotes the presence of proteins in the sample. Coming to the Ninhydrin test, to 5 ml of the protein sample, add 0.5 ml of Ninhydrin reagent and heat in the water bath. We can see the purple color formation, which denotes the presence of free amino acids. The free amino acids present in the proteins can be easily identified by this Ninhydrin test. Next is about the coagulation test. To about 2 ml of the protein sample, add few drops of concentrated HCl. A coagulation will be formed. This denotes the presence of proteins present in the sample. Next is about the Molish test. To 5 ml of the protein sample, add few drops of Molish reagents and add concentrated sulfuric acid along the sides of the test tube. A green coloration is obtained. If the green color is obtained, then this denotes the presence of glycoproteins present in the sample. About the xanthoprotic test, to 5 ml of the protein sample, add few drops of concentrated nitric acid and then heat in the water bath. Next, cool it and add sodium hydroxide. At first, it forms white color precipitate. Upon adding sodium hydroxide, it will form a yellow color precipitate. This denotes the presence of amino acids with aromatic nucleus. So we can see this color of yellow coloration upon adding sodium hydroxides. Okay, coming to the conclusion, we have done the test and we have confirmed the presence of protein sample. First test is about the solubility. To the protein sample, when we added cold water, we can see the insolubility, and when we added hot water, we can see the least solubility. Second test, to the protein sample, when we add HCl, we can see the coagulation. And about the confirmatory test, Biuret test, we can see the violet color formation when we added Biuret reagent, and this confirms the presence of peptide bonds in the protein sample. And finally, about the nin-hydrin test, when we added nin-hydrin reagent to the protein sample, we can see the formation of purple color. This denotes the presence of free amino acids present in the bond, the protein sample. Likewise, we are having some more tests like xanthoprotic, Molish test, nitric acid test, by which we can confirm the presence of protein samples. So these tests are helpful in finding the presence of proteins in any samples, like in foods or in blood samples or in any other research samples. We can use this test and we can find whether the proteins are present in our body. Thank you.